Okay. Yeah. Good morning, Ms. Chwakendi. My name is KARTHIRAJ. Today we are from Biomedical Science Batch 2 Year 1. So today I'll be explaining about antimicrobial resistance. So my name is KARTHIRAJ and my colleagues are SHARVEENA, Dhaneesh and PRASHVINA. Next slide. So I'll be explaining about what is antimicrobial resistance. So antimicrobial resistance refers to the ability of microorganisms such as bacteria, viruses, parasites, and fungi to withstand the effect of antimicrobial drugs, including antibiotics, antiviral, antifungal, and antiparasitic medication. So it occurs when the microorganism adapt and develop mechanism to resist the drugs that were once effective in treating them. So AMR is a global health concern that possesses a significant challenge in the prevention and treatment of infection disease. When microorganism becomes resistant to antimicrobial drugs, infections can be more difficult to treat, leading to prolonged illness, increased healthcare costs, and higher rates of morbidity and mortality. The development of antimicrobial resistance is primarily driven by the inappropriate or excessive use of antimicrobial drugs. So there are several factors that contribute to AMR, including misuse of antibiotics in human medicine, overuse of antibiotics in agriculture and animal husbandry, inadequate infections prevention and control practices, and the lack of access to clean water and sanitation facilities. So the consequences of AMR are far-reaching. It can render previously effective treatments to ineffective. It can increase the complications and treatment failure, prolong hospital stays, and escalate healthcare costs. It also limits the effectiveness and routine medical procedures such as surgeries, chemotherapy, and organ transplantation, which we can rely on effective antimicrobial drugs to prevent and treat infection. Next slide.
What is viruses? Viruses is an infectious disease caused by the bacteria Burkhardia pseudomonas. It is primarily found in Southeast Asia and Northern Australia. Although cases have been reported in other parts of the world, the bacteria is commonly found in soil and water and can infect humans and animals. Burkhardia pseudomonas is a gram-negative bacterium and causative agent of melioidosis, a potentially severe infectious disease. It is commonly found in soil and water in tropical and subtropical regions, particularly in Southeast Asia and Northern Australia. The bacterium has also been identified in other parts of the world, including areas with similar climates. What is Burkhardia pseudomonas? It is an opportunistic pathogen that can infect both humans and animals. It enters the body through the direct contact with contaminated soil or water. Inhalation of aerosol particles or rarely through injection or through wounds and cuts. Viruses caused by pseudomonas can manifest in various forms, ranging from localized skin infection and excessive to severe systematic infections that can affect multiple organs. The symptoms of melioidosis can be diverse and non-specific, making diagnosis challenging. Common symptoms include fever, cough, difficulty breathing, skin abscess, joint pain, and septicemia.
I'll be explaining the mechanism of melioidosis. So, melioidosis is caused by bacterium Burkhardia pseudomonas. It is a mechanism involved in various stages and infection and interactions, which the host immune cells system. Here are the general overview of the mechanism of melioidosis. As you can see here, there are a simple diagram how a bacteria infect a healthy cell to multiply its bacterium. So the first one is entry and infection. So Burkhardia pseudomonas can enter the body through different routes, such as inhalation, ingestion, or direct contact with contaminated soil or water. Once inside the body, the bacteria can invade various cells and tissue, including epithelial cells, macrophages, and other immune cells. The second one is adhesion and colonization. Burkhardia pseudomonas possesses various surface structures, such as adhesives and pili, which allow to adhere to colonize host cells. The bacteria can then establish infections in multiple organs and tissues, including the lungs, liver, spleen, and kidneys, which are the main component for our body to work. The third one is intracellular survival and adaptations. Bucharodius pseudomyelitis has evolved strategies to survive within the host cells. It can evade or resist the killing mechanism of immune cells, including phagocytes. The bacterium can replicate and survive inside phagocytes, enabling them to avoid detection and destructions. The fourth one is virulence factor. So Bucharodius pseudomyelitis produces several virulence factors that contribute to its ability to cause disease. These factors include exotoxin, enzymes, and proteins that help the bacteria survive and manipulate the host immune response. The fifth one is inflammatory response. So the interaction between Bucharodius pseudomyelitis and the host immune system triggers an inflammatory response. So these immune systems release cytokinins and chemokinins to recruit immune cells to the site of infection, leading to the formation of granulomas. So the sixth one is, as we can know, is granuloma formation. So granulomas, which are aggregates of immune cells, forms a protective mechanism against Bucharodius pseudomyelitis. So these structures attempt to contain the infection and limit the spread of bacteria. However, the bacteria can persist within the granulomas and cause the chronic infections. The seventh one and the last one is systemic dissemination. In some cases, Bucharodius pseudomyelitis can escape from granulomas and spread throughout the body via the bloodstream, leading to a systemic infections. This dissemination can result in severe complications such as sepsis and multiple organ failure. Next slide.
I'll talk about the transmission. Maridiasis is primarily transmitted to humans through direct contact with contaminated soil or water that contains the bacterium Bucharodius pseudomyelitis, the causative agent of the infection. The most common routes of transmission include skin contact. The bacterium can enter the body through cuts, abrasions, or open wounds when they come into contact with soil or water containing Bucharodius pseudomyelitis. Occupational exposure, such as in agriculture workers or those involved in soil excavation, increases the risk of infection. Number 2 is inhalation. Inhalation of aerosolized particles contain Bucharodius pseudomyelitis, particularly during activities that dissolve contaminated soil or water, which can lead to respiratory tract infection. Number 3 is ingestion. Consumption of contaminated water or food, especially in endemic areas, can be a rare route of transmission for maladies. Person-to-person transmission of maladies is rare, but it can occur in specific situations. It usually requires close and direct contact with the body fluids or secretion of an infected individual. Transmission can occur through activities such as sexual contact or via respiratory droplets when an infected person coughs or sneezes. Next slide. Signs and symptoms. Maladies can present with a wide range of symptoms, and the severity of the disease can vary from mild to severe. Some individuals may have no symptoms at all, while others may experience a combination of signs and symptoms. The onset of symptoms can occur anywhere from a few days to several months after exposure to the bacterium Bucharodius pseudomyelitis. Common signs and symptoms of maladies include: the one is the localized infections, where the skin infections, development of skin ulcers, episodes or sores at the site of entry, often resembling boils. Number 2 is the pneumonia. Symptoms can include fever, cough, chest pain, and difficulty in breathing. Maladies-related pneumonia can be severe and life-threatening. Number 2 is the systematic infections, which is fever. High fever is a hallmark symptom of maladies and is often persistent or recurrent. Number 2 is generalized weakness and fatigue and malaise. Respiratory symptoms is cough, shortness of breath, and chest discomfort. Joint pain, the pain, swelling, and stiffness in the joints take place. Abdominal symptoms, where abdominal pain, nausea, vomiting, and diarrhea. Urinary symptoms, where urinary frequency, urgency, and pain or discomfort during urination. And septicemia, where severe cases can progress to septicemia with symptoms such as rapid breathing, confusion, and low blood pressure. Next slide.
I'll be explaining of risk of exposure. So the risk of exposure to melioidosis varies depending on geographical location and specific environmental factors. So melioidosis is considered as endemic disease in certain regions, particularly in Southeast Asia and Northern Australia, where the bacterium Buchardia is naturally found in soil and water. So there are factors that can increase the risk of exposure to melioidosis include geographical location. The highest incidence of melioidosis occurs in regions where Buchardia is endemic. So this includes the countries such as Thailand, Malaysia, Singapore, Vietnam, Cambodia, and parts of Australia. The second one is occupation. Certain occupation may increase the risk of exposure of Buchardia pseudomyelitis. So this includes agriculture workers, construction workers, and individuals who involved in activities that require contact with soil or water, such as rice farming, digging, or excavation work. The third one is environmental conditions. Melioidosis is more prevalent in areas with specific environmental conditions, such as tropical or subtropical climates and high rainfall. Soil disturbance caused by heavy rain, flooding, or construction activities can increase the release of the bacteria into the environment. The fourth and the last one is immunocompromised individuals. People with weak immune system, such as those who have diabetes, chronic lung diseases, kidney disease, or HIV/AIDS, have higher risk of developing severe melioidosis if exposed to Buchardia pseudomyelitis. Next slide. The next one is prevention. Prevention of melioidosis involves minimizing exposure to the bacterium Buchardia pseudomyelitis, which is primarily found in the soil and water in certain regions. Here are some preventive measures where the individual can take. The first one is personal hygiene. The individual must practice a good hygiene, including the regular hand washing with soap and clean water, especially after handling soil or engaging in activities that involve potential contact with contaminated environments. The second one is wear protective clothing. When working or participating in activities that may involve contact with soil or water in melioidosis endemic areas, we must wear appropriate protective clothing such as gloves, boots, and long sleeve shirts to minimize the direct skin exposure. The third one is safe water consumption. Drink safe treated water from reliable sources. Avoid drinking untreated water from potentially contaminated sources such as stagnant ponds, puddles, or untreated well water. The fourth one is avoid high risk environment. So we must minimize direct contact with the soil and standing water in areas known to be endemic of melioidosis. And we also must avoid wading or swimming in muddy or contaminated water sources. The fifth one is wound care. Clean and protect any cuts, abrasions, or open wounds with clean bandages or dressings to prevent the potential entry of bacteria. Sixth one is insect bite prevention. So we must protect ourselves from insect bites by wearing long sleeves, using insect repellents, and sleeping under bed nets if necessary. Although melioidosis is not primarily transmitted by insects, but preventing other insect-borne diseases can help to maintain our overall health. The seventh and the last one is awareness and education. We must stay informed about melioidosis and its risk factors, especially if residing in or traveling to endemic regions. We must be aware of the signs and symptoms of the disease to seek medical attention promptly if necessary. It's important to note that while these preventive measures can reduce the risk of melioidosis, they do not provide complete protections. Individuals with specific risk factors such as diabetes, chronic lung disease, or compromised immune system should have extra precaution and consult healthcare professionals for personalized guidance. Additionally, at the community level, public health measures may include surveillance of melioidosis cases, proper water disposal, and regulation of activities involving handling of soil or contaminated water sources. Next slide.
Talk about the treatment. The treatment of melioidosis involved a prolonged course of antibiotics to eliminate the bacterium Brucordia pseudomyelitis from the body. The specific choice of antibiotics and the duration of treatment depend on the severity of the infection, the site of infection, and the patient's overall health condition. Treatment may involve, number one is the intravenous antibiotics. In severe cases or when the infection has spread to multiple organs, intravenous antibiotics are typically administered in a hospital setting. Commonly used intravenous antibiotics include ceftazidime, meropenem, imipenem. These antibiotics are generally given for an extended period, often ranging from 10 to 14 days or more. Number two is the oral antibiotics. Following the initial intravenous antibiotic treatment, a transition to oral antibiotics may be to make complete the course of treatment. The most commonly used oral antibiotic is trimethoprim sulfamethoxam. This step is crucial for long-term suppression of the infection and prevention of relapse.
Very good day to the respected lecturer and to my fellow friends. Me, Dhaneesh and I will be presenting on another disease that is associated with antimicrobial resistance, Neisseria gonorrhea. So, what is Neisseria gonorrhea? It is a sexually transmitted disease that is caused by a bacteria known as and it usually targets individuals from of 19 to 24 and individuals with active sexual life. The disease could be treated early if it at an early stage and could potentially be treated well. And if it's not, transmit a deadly virus known as HIV that could cause AIDS. So there are a few symptoms of gonorrhea in both men and women, and the symptoms varies for each gender. So for most men, they stay asymptomatic, and but there will be present of some signs of urethral infection that includes is a painful urine infection. And apart from that, white or yellow or white discharge are also present in men. That some men also experience testicular. So moving on to the symptoms of the disease in men. Symptoms of Neisseria gonorrhea infections are most women stay asymptomatic, similar, but they will be present with mild symptoms that they often mistake with other diseases. Some other symptoms are dysuria, abdomen, and increase in vaginal discharge. The discharge could be brownish or even white discharge could be seen. In addition, some women also experience vaginal bleeding. It's not period blood, but it is the bleeding that is caused by bacteria itself. Next, the bacteria produces rectum and affects bowel movement in females.
Hi, my name is Dhaneesh, and I would like to talk about mechanisms of Neisseria gonorrhea in male and female, and I would like to talk about the treatments as well. First, I'd like to talk about male. Entry and invasion and colonization, attachment and penetration, inflammation and symptoms, and also systemic dissemination. So, Neisseria gonorrhea infection of men most commonly occurs as an acute urethritis, which develops from the concomitant inflammatory response directed at infecting gonococci. However, a small percentage of men will develop asymptomatic gonococcal urethritis. One hallmark of symptomatic gonococcal disease in men is the presence of a purulent discharge, which is associated with PMN influx and shedding of urethral epithelial cells. Human volunteer studies indicate that there is an incubation period from the time of infection on the set of clinical symptoms of disease. During this time, gonococci cannot be cultured from the urethra up to 40 hours after the initiation of the infection, after which a purulent exudative process begins. These data suggest that gonococci enter a protective environment early in disease where they survive and that infected epithelial cells are subsequently shed from the mucosal urethral epithelium. The release of cytokines and chemokines from the urethral epithelium may potentially initiate the inflammatory response associated with gonococcal urethritis by triggering PMN influx. Studies performed by using the human challenge model of gonococcal disease have demonstrated that the ability for gonococcal to acquire iron from its sole human host is required for successful colonization of the male urethra. Now I'm going to talk about women, which is female, lower tract of tongue. In contrast of the inflammatory response generated predominantly with gonococcal infection of the male urethra is 50 to 80% of women with lower genital tract infection are asymptomatic, and 70 to 90% of women with this symptomatic infection lack signs of genital tract involvement. Analysis of cervical secretions obtained from normal, which means uninfected women, and from women infected with gonococcus also reveal that an antibody response is not generated with uncomplicated infection. These clinical findings are consistent with the ability of the gonococcus to evade and subvert host immune function. Upon infection of primary cervical epithelial cells, gonococcus release a phospholipase D homolog that gains access to the cervical intracellular environment, not specifically though micropinocytosis of gonococcus. Ascending infection of the uterus and fallopian tubes may occur as a consequence of hormonal changes that alter the mucosal epithelia, molecules available for gonococcus use, or virulence factors expressed by the gonococcus. Next slide.
There are a few preventative measures that transmission with personal hygiene. All of us should have a personal showering daily, then use the environment that secondly, we are expected and it is advisable for appropriate certain places and areas that involves much physical activity, as boots and gloves. Thirdly, safe water consumption. It is highly recommended to follow water filter or ponder that we use to avoid the contamination of the to avoid high-risk environments. We should prevent the exposure of ourselves to bacteria and wear masks to avoid transmission from infected. Fifth, if there's presence of any treat it immediately. Professionalism find the invasion of bacteria onto insect bite prevention. We should use repellent or any other medicine to prevent ourselves from insect bites. All of us, it is well known that the bacteria could be transmitted also by insects, so it is advisable to use repellents, any other insect repellents. Last but not least, awareness and education. Parents, media authorities, and other respected authorities such as and governments should spread awareness to younger generation by giving them a proper education. Transmission. It is being safe because to prevent the this and to create this healthy young generation.
So I would like to talk about diagnosis and also treatment for Neisseria gonorrhea. So I'd like to go for the diagnosis first. To determine whether you have gonorrhea, doctors will analyze a sample of cells. The samples can be collected by urine tests. This can help to identify the bacteria in your urethra. Moving on, swab of affected area. The swab of your throat, urethra, vagina, or rectum can collect bacteria that can be identified in lab. Now, I'd like to talk about the treatments. There are three types of treatments can be undergone for adults, babies, and partners. For adults with gonorrhea are treated with antibiotics due to emerging strains of drug resistance. Neisseria gonorrhea, the Centers for Disease Control and Prevention recommends that uncomplicated gonorrhea can be treated with the antibiotic called ceftriaxone given as a shot called an injection. After getting the antibiotic, he could still spread the infection to others for up to 7 days. For babies born to mothers with gonorrhea who develop the infection can also be treated with antibiotics. So now, for partners, your sexual partner or partners from the last 60 days also need to be screened and treated, even if they have no symptoms. If you're treated for gonorrhea and they aren't treated, you can become infected again through sexual contact. Make sure to wait until 7 days after your partner is treated before having any sexual contact. Next slide. So that's all from us. Thank you very much.